welcome to our final Masterclass of the University of Melbourne's 11th Annual Science Festival as part of National Science Week. My name is Julia and I will be your host once again for today. As the host, I'd like to acknowledge the traditional owners of the unceded lands on which we are filming, the Boonwurrung people. Aboriginal and Torres Strait Islanders are this country's first scientists, with deep and enduring knowledge of the land, waters and skies. I pay my respects to elders, past and present, and I welcome all First Nations people here today. In making this acknowledgement, we commit to respectful and responsible conduct towards all others according to the traditional laws of this land, particularly at times of formal ceremony. So today's event, our final event, is How Antibiotic Resistance in Animals Impacts Public Health with Laura Hardefeldt. Today we are coming to you live from our Werribee campus, a hub for teaching, research and equine specialist care. And it hosts the Werribee Equine Centre and Green Cross Veterinary Hospital. Here, the next generation of veterinarians and veterinary researchers are taught. We also, in the room today, have students here from Alamanda College. So welcome all the Year 9s today. So how today will work, as it has worked all week, Laura, our presenter, will be giving a 30-minute presentation about her work, and then we'll be taking questions from our audience. And then aim to finish up about 11:30. Now, to ask a question for all of you at home, the Q&A function is open on Zoom, so put all your questions in there throughout the presentation and we'll get to as many as possible at the end. For all the students in the room, when we get to the question and answer time, just put up your hand and we'll run around with a mic so everyone at home can hear you as well. Now, there will be polls throughout this presentation, so everyone at home, just wait for those to pop up, and we'll be including everyone in the room as well with some of those questions. Now, finally, the chat is also open for the Zoom, so we welcome all comments. Clapping, cheering emojis are also very much welcome, but of course, please be respectful. Now, to make sure everything is working at home, could I ask you all to open up the chat function and let us know where you are Zooming in from, whether it's your suburb, whether it's your school. I know there's a lot of school groups that do join us each session, or maybe it's the country you're Zooming in from. If it's not Australia, I know people from all over the world do join us for these masterclasses. So open up the chat, pop in where you are Zooming in from so we can find out where everyone is from. All right, everyone at home hopefully can see where everyone is from. So welcome and thank you for joining us. So without further ado, all the intros are done. I would like to welcome Laura to come on in and give her presentation. And I believe I'm going to be handing around some first experiment.
Yes, thanks, Julia. So as part of the activity today for everybody who's joined us here in the class, I've got these two bowls. So if you have a pet that comes inside your house, please take one out of the red bowl. If you have a pet that sleeps in your bed or somebody else in your family's bed, then please take a pet out of the red bowl. And at the end of the class, we will chat about it'll lead to some chat at the end. So Julia's going to pass those around for me. Thank you very much. So today we're going to talk about antibiotic resistance or superbugs. And antibiotic resistance, bacteria are present all over the world, everywhere. Every surface that you look at is covered with bacteria. So obviously bacteria can grow in the laboratory on those little petri dishes, and you can see there a whole bunch of different bacteria. You can tell they're different bacteria because they're all different colours, they're different shapes, but there is bacteria in the food that we eat. There's bacteria in our food animals. So the sheep there are from the farm that I grew up on in central Victoria. There's bacteria in the super wild environments. So the icebergs there are in Alaska, and you'll find bacteria covering those, but also in the wildlife that live in those environments, in our urban environments, and of course also on our kids and on our pets. So, and when bacteria become resistant to antibiotics, simple infections can become chronic, serious, or even life-threatening. So there was some work done by the government in the United Kingdom a few years ago now that looked at the risk that antimicrobial resistance is going to pose to the global population of people over the next coming decades. And they found that in 2014, there was about 700,000 people each year dying of antimicrobial resistant infections. But by 2050, that the number of people who die is going to be, it's going to be the largest cause of death. So over 1.5 million people will die of antimicrobial resistant infections every year. And so it's going to cost the global economy, but also it's going to cause issues with like things that we take for granted now. So things like people getting chemotherapy, people getting joint replacements will all be complicated by the fact that there's these bacteria that are resistant to lots and lots of different antibiotics. So if we think about how bacteria become resistant to antibiotics, there's a few different ways that that can happen. So one of the simplest ways is if you think in the first Petri dish there, we've got a whole bunch of susceptible green bacteria. And as bacteria replicate, then they can undergo mutations in the genes, and just by random chance, you can get a mutation that leads to resistance against a particular antibiotic. So that's just a random thing. And these can come and go in the environment, and it's not such a big deal unless we put in a selective pressure. So if you add an antibiotic to that environment, all of a sudden, all of the bacteria that are susceptible to that antibiotic are going to be killed, but the resistance one has a survival benefit there. And so it survives. And if we keep that antibiotic in the background, then that resistant bacteria can grow and it becomes the dominant population. So you can imagine if we had a sink here that was full of antibiotics, the only bacteria that would be able to grow in that environment is a bacteria that is resistant to that antibiotic. And it turns out that bacteria are really good at getting resistance. And that's because most of the antibiotics that we use in medical practice and in veterinary practice these days come from the environment. So the first one came when they were growing a fungi on a plate, penicillin, and they realised that that fungi was killing the bacteria that surrounded it. And so that is how they discovered penicillin. And most of the other antibiotics that they've discovered since that time also come from the soil or from the environment in some way. And so bacteria are really good at developing mechanisms to avoid being killed by these environmental agents. And the same happens in when they're exposed in a clinical situation. And it turns out this process is really old. So people have looked in the permafrost from the Arctic region, so from soil that's kind of 50,000 years old, and found antibiotic-resistant bacteria in that environment. So this is not a new process. It's been going on for a really long time. But one of the interesting things that bacteria are also very good at doing, not only developing resistance, but also sharing resistance. So if pretend I'm a bacteria and I have become resistant, I can share my resistant genes with you. So you don't have to develop it yourself. I can just share it, and then you become resistant as well. So you can see how resistance can spread between bacterial populations as well as in a lineage of bacterial populations. And so that's why antimicrobial resistance is becoming such a big issue, or superbugs is becoming such a big issue. And this process can happen really quickly. So we're going to play this video that shows just how fast it can happen.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
Right. So Julia's going to jump in here because we've got a couple of poll questions.
Hello, hello. Okay, now I'm still actually blown away by that video, but that was amazing, so I need to focus on this poll. Okay, so everyone at home, there is a poll coming up, and it should be up on your screen now. So have you ever had a course of antibiotics? 3 options: never, yes once or twice, or yes multiple times. So pop that into the chat and that should appear soon.
Okay, and for everyone in the room, I'd like you to stand up if you've ever had a course of antibiotics.
And I'll stand up myself.
Me too.
Okay, that's most people.
Yep. So we've probably got about 70%, 80% of people in the room that are standing up. What about on the poll?
This is still waiting to be closed and opened up. Oh, yeah, coming up. Okay, never, only 11%, 26 once or twice, but the majority 63%, multiple times.
Yeah. Yep. Okay, so let's stay standing if you have had a course of antibiotics in the last year.
Oh.
And for our online...
I would sit down for that one.
We've got a poll question on this online too.
Yep, that should be coming up.
So we're probably down to about half of the people in the room still standing.
Okay, so in the last year, so no, actually 78% of the audience has not had a course of antibiotics. Yes, 1 or 2 times, 22%, and none for multiple times.
Yeah.
So there you go,
Great.
Very good.
So the research would suggest that half of Australians, the general population of Australians, have an antibiotic. You can sit down now.
Okay, all right, and I'll leave you to it.
Thanks, Julia. Have had an antibiotic in the last 12 months. So Australia is one of the highest users of antibiotics in the general population out of anywhere in the world. And we use a lot of antibiotics in our animals as well, in our pets. So our antibiotic use in our pets is about 1 in 5 dogs and about 1 in 10 cats get exposed to antibiotics in any year. So we use a lot of antibiotics. But if we just take a step back and think about why antimicrobial resistance is becoming such a big problem, why are superbugs becoming a big problem? And I think there's 3 major reasons for that. The first is that we're exposing the bacteria in our environment to a lot more antibiotics than there has been in the past. So we really, as a society, rely on antibiotics a lot. And we also live in a very globally connected society. So 100 years ago, there was not many people moving around the world. And so if we had resistance develop in Australia, there was a pretty low risk that it would get transferred to another country. And you could even say that for a city. So if we had resistance develop in Melbourne, it was unlikely, you know, to get transmitted to another more remote part of Australia. But that's changed, right? We travel all over the world and people are moving around an enormous amount. And so in places where antimicrobial resistance is a much bigger problem than it is in Australia, like say in India, for example, there's a lot of antimicrobial resistance in India, then if you visit India, then you can bring those antimicrobial resistance infections back to Australia. And we know that that's a huge problem. Especially if you come back and get sick, then you're more likely to be sick with one of those sorts of infections. The other problem is that we take a lot of antibiotics when we don't need them. And people do that. They treat their animals for viral infections. And I guess the most common one and the one that jumps to everyone's mind is when you take antibiotics when you've got a cold. All of our colds are caused by viruses. Antibiotics don't treat viruses. They only treat bacteria. And so when we take antibiotics when they're not needed, then we're exposing all of the bacteria in our bodies, in our gastrointestinal tract. Bacteria live in our lungs, everywhere. They're living all over your body, in your body. Then we expose them to antimicrobial resistance. And any antibiotic use can lead to antimicrobial resistance. So, how many people have a pet that lives inside with them? We'll bring Julia back in.
Yeah, I know. There's going to be another poll, everyone
Yes.
at home. So that's going to pop up. Okay, so do you have a pet? Okay, have you got 4 options? No? Or 3 yeses. Yes, outside only. Yes, comes inside the house. Or yes, sleeps on my bed. Very different boundaries.
So let's, for people in the room, while we're waiting for the online poll, stand up if you've got any pets at home.
I'm sitting down.
I'm standing up. Okay, so probably about half the people in the room have a pet. Stay standing up if your pet comes inside. I think that's everyone.
Oh, yeah,
everyone who's got a pet comes inside. Stay standing up if your pet sleeps on a bed in your house. Yeah, so probably most people have a pet sleeping on their beds. Yep, great. What about online?
Okay, online. Oh, okay, bit of a mix. So, yeah, no pet, similar to inside. There was 0% yes, outside only. 29% both for yes, comes inside and yes, sleeps on the bed.
Yeah.
And then no pet at all was 41%.
Okay, so that's pretty representative of the situation in Australia. About half of Australian households have a pet.
Very good.
And of people who have a pet, about half of them, 60-odd%, have pets that sleep on their beds.
Well, there you
them.
go.
So
Very
there
good test audience there. Very good. All right, I'll leave you to it.
Great, thank you. And if you think about bacteria, we look at this picture in a whole different light now, because the bacteria, who just raise your hand if you have a pet, if this would be a situation in your household. Yep, so lots of people have pets that lick them on their faces. My dog licks me all the time, drives me crazy. But it's a really common scenario, right? Like, our pets are part of our family. They're really important to us. And they do live very intimately in our households. And so this is not an uncommon scenario. But if we think about the bacteria from a bacterial perspective now, you can see how bacteria move around the household. And we know from quite a lot of big breadth of research now that the bacteria that you have on you gets transferred to your pets, and the bacteria that your pets have on them and in them gets transferred to you and your environment as well. So all of we share the bacterial community around our household. And what about antibiotic use outside of our households? So antibiotics are used for the production of food. So in Australia, we're really lucky in that we don't use so much antibiotics in our food animals as in some other parts of the world, which has got to do with a few different reasons. But the first is that most of our animals are grown in very big paddocks, not in really intensive environments. So if you, for example, are living with, if we said this whole classroom here was living in this small environment all the time, you could see how bacteria and infections would pass between you very easily because you're all in really close contact. But actually, we all live in separate houses, and so the risk of bacteria passing and infections passing between us is much lower. And the same happens with animals. If we introduce 1,000 animals into this small room, there's going to be lots of infections that pass really easily. But actually in Australia, our dairy cows and our sheep and our beef cattle all live in very extensive environments, and so there's much less risk of infection and much lower antibiotic use. So we're very lucky in Australia from that perspective. But we do also use antibiotics on our crops. So food is often treated with antibiotics to stop infections that might ruin our fruit crops, for example, or our grain crops and that sort of thing. So some other environments where antibiotics are used. And anytime we use antibiotics, we're selecting for antimicrobial resistant infections, for superbugs. But most antimicrobial resistance genes are lost in the population over time. So if we think about our superbug, it acquires antimicrobial resistance genes to protect it from the antibiotic. But that's really, it's a heavy thing for it to carry around. So imagine that you are going into battle and you're wearing an armour and you're carrying lost the word now, a shield. Thank you, Julia. Then, you know, that's a really good thing to have while you're in battle, right? Because it protects you. But once the battle's over, you don't want to be wearing your armour and carrying your shield around when you're going about your daily life, because it's really heavy. It's exhausting. And the same is true for bacteria. So carrying around these resistance genes is really hard work for them. And in general, over time, they stop carrying them. They get rid of those genes. They don't need them anymore. The battle's over. And so they go back to being a susceptible population. And so in general, that kind of takes around sort of 3 to 12 months, usually around 6 months. By about 6 months, the bacteria in your gut have gone back to being the normal population before you took antibiotics. So that's a really important concept as well. So what can we do to prevent antimicrobial resistance being a problem? So the first thing to do is to protect yourself from bacterial infections. So really simple things like washing your hands. We know that washing your hands for 20 seconds with soap is, as just normal everyday soap, is a really good way of getting rid of the bacteria on your hands. So after you have touched your pet, after you've been outside in the environment, it's a really good idea just to wash your hands when you come inside, particularly before you eat, because putting things in your mouth is a good way of exposing your body to antimicrobial resistance. The other really important way of protecting us and also protecting our pets against infections is to get them vaccinated, to be vaccinated against the common infections that we're exposed to. And then if you don't get sick, then you're not likely to need antibiotics. So we can reduce the use of antibiotics by protecting ourselves against infections using vaccines. The other really important thing to do is to not take antibiotics when you don't have a bacterial infection. So if you've got a viral infection, then going to the GP and getting antibiotics is not going to make you better any faster. And the same goes for our pets. We know if they've got a viral infection, if they've got an infection that is self-limiting, then treating them with antibiotics is not going to make them better any faster. And we know as pet owners, what we want is for our pets to get better fast. And your parents, they want you to get better as fast as possible. And so that leads to this use of antibiotics because everyone just wants to feel better faster. It's no fun seeing your pet being sick, but it's really important to not pressure your GP or your vet to give antibiotics when they tell you that they're not needed. So those are some really simple ways to reduce the impact of antimicrobial resistance or superbugs in you and your family. And now we're up for some Q&A.
Amazing. Thank you so much. Everyone, round of applause for Laura. Thank you so much for the presentation. Fascinating and so many really interesting concepts there. We've already got quite a few questions coming in online, which is excellent. So yes, a reminder to everyone, so just open up the Q&A function on Zoom and pop in your question and we'll get to as many as possible. And of course, everyone in the room as well, you've seen quite a bit of things and I'm sure you've got some questions. So pop up your hand, we've got a mic. Taylor's going to be running around with the mic and we'll try and maybe alternate in the room and online. While I'll give everyone a bit of thinking time for some questions. So I might just kick it off with, I guess, asking about your background. So I guess what did you study and what led you to this area of research?
Yeah, so I'm a veterinarian and I worked in clinical practice for 15 years, mostly looking after horses. And then I was interested in antibiotic resistance. We were seeing more of it in our clinical practice and interested in both the behaviour around prescribing and also the antibiotic use itself and antimicrobial resistance. And so I did my PhD here at the University of Melbourne and have stayed on as a researcher in that field.
Amazing. Yeah. Fascinating. I'm sure there's a lot of people out there that considering a similar pathway, so I'm sure they might have more questions. Okay, so I might start with some online. So the first one comes from Beck. So if antibiotics become ineffective, are there any other ways we can treat bacterial infections? Any historic techniques that we might be able to resort to?
Yeah, great question. And there's quite a lot of research going on into this at the moment. So the number of new antibiotics that are being developed is really small. So you might see in the newspaper occasionally that they've discovered a new antibiotic peptide, there was one recently that came from Tasmanian devil milk. They found an antibiotic peptide in Tasmanian devil milk. Sure. But unfortunately, very few of these agents ever come through the pipeline to become an antibiotic. There's a few different reasons for that, but in general, we're not getting new antibiotics. And so lots of people have been looking into alternative ways of treating bacterial infections in people. And there's some really fancy kind of phages that are viruses that attack bacteria that have been looked at. But also things, really simple things like honey for wounds and other alternatives to antibiotics as well. So a very active area of research, I would say.
Yeah, absolutely. Okay, so we've got one question from the audience. Please take it away.
I noticed in your... Hello. I noticed in your earlier diagram that the bacteria that's resistant to antibiotic is in lower populations. There's only one of the red one, and there's many of the green one. Is there a reason why bacteria that's resistant to antibiotic don't thrive in a normal environment?
Yeah, so in general, it is quite a burden for bacteria to carry resistance genes. And so they do tend to lose them in the population over time. And it's not always the case. Sometimes they do hang on to those genes for a long period of time. But in general, bacterial populations tend to go back to being susceptible over time because it is a fitness cost to carry those resistance genes.
Interesting. Good question. Okay, we'll go one online. So from Magdalene, is viral resistance to antivirals also an issue?
Less of an issue. So the way that viruses respond to antivirals, it is less of an issue than antibacterial resistance. We also see antifungal resistance, which is a bit of a problem in people, less of a problem in animals. So less of those drugs used in the population compared to antibiotics. But yeah, there are issues in other microbes as well as bacteria.
Yeah, interesting. Okay, any more questions? I'll go one more online, I think, and maybe give some more time for everyone in the room. Okay, one from Holly. Does having pets inside build up our immune system? Holly says, I'd like to think that they're just family germs, especially when my dog and toddler kiss each other. Thanks for the extra info, Holly.
Yes, so the earlier picture of the kids on the couch with the dog is my kids with my dog. But yeah, there is really good evidence that having pets, especially around kids, is good for your immune system. But I think we just need to be cognizant of some of the downsides as well, especially if we have if we've got healthy pets and healthy kids, then the risk is probably pretty small. But if you have somebody who's immunocompromised, so if they're on chemotherapy, for women who are pregnant, for elderly people, and especially if you have a pet that's been on and off antibiotics a lot, then the risk might be higher, and so we just want to be really careful in those scenarios that we're not causing harm.
Yeah, absolutely.
Yeah.
Okay, maybe just a quick question for everyone in the room. Is anyone thinking of becoming a vet? Maybe hands up. Oh, one or two, okay. Maybe. Well, I was just going to say, because obviously we've got a vet in the room, so maybe think about some other questions you could ask Laura about maybe her journey into becoming a vet and or research as well. So just have a bit of a think. Okay, so back online. So Jessica, how do you do AMR research in pets and livestock? With human antibiotics, we do the research in mice. Would that be translated well to other animals, or do you do preclinical in animals other than mice straight away?
Yeah,
Come on, Jessica. Yeah.
So we do a lot of. My research group does a lot of clinical research. So we collect bacteria from sick animals and also from healthy animals. So you can collect bacteria by doing a swab of the skin or by looking in animal poo or by taking a swab of the mouth. So lots of different samples we can take from healthy animals and looking at the resistance that's there in a healthy population versus that's there in a sick population. And also I'm really interested in how long that resistance lasts and what impact it might have not only on the animal that's being treated, but also by its friends that are living around it. So we do some of that research in horses, looking at,
Nice. Well, sticking on, I guess, like the field work and all the research you're doing, maybe do you want to talk through sort of a recent sort of research in the field that you did and sort of the steps that you went through, I guess, to explain, to everyone in the room and online what that process is.
Yeah. So I'll tell you about this project that we just did looking at resistance in horses. So we were interested in how horses, the bacterial population in their gastrointestinal tract and also in their lungs changes when they change environments and also when they're exposed to antibiotics and when they're not. So we had 100 horses that were living on 3 different farms and they were all kept in slightly different managed in slightly different ways. So there was one group of about 30 horses that were treated quite a lot with antibiotics. There was one group that about a third of the horses on that farm had been exposed to antibiotics, and there was another group that had essentially no antibiotics at all. And we took samples we took swabs from their poo and also from the back of their throat, before they came onto these farms, when they were about halfway through the study period, so after about 3 weeks, and then at the end of the study period, about 6 weeks. And we are looking at the amount of resistance that they got in their lungs and also in their gastrointestinal tract over time. But also what we're really interested in is if you were treated with antibiotics, Julia, and I was standing in the stall next to you, do I get those resistance genes as well? And does it impact the other bacteria in my gastrointestinal tract? And some of the really interesting preliminary findings is that so all of you guys, before you came onto the farm, if you were all of our horses, you all had very different bacteria in your gastrointestinal tract. So you all looked really different. But then within that first period of time, within the first 3 weeks, pretty much you all came together and developed the same bacteria. So within a really short period of time, you all had very similar bacteria in your gastrointestinal tract. And so we can see how diet and environment and stress and that sort of thing can really influence the bacteria that you have. And now we're looking at what resistance is there on farms that didn't use much antibiotics, used a bit and used a lot.
Yeah,
so we'll be able to look at the differences. Yeah.
Amazing. I mean, big research project. Working with 100 horses, it's very different to much smaller animals. How did you connect in and sort of recruit, I guess, 100 horses? And I guess obviously the farmers in that.
I guess that's where having worked in equine practice for a long time is really helpful. So I know a lot of veterinarians and we were able to recruit farms through and kind of get our vet friends to help us to collect these samples and spread the load, I guess. Yeah,
so, absolutely. Yeah. Well, I guess, the benefits obviously would, that they'd see them as well on the farm. So everyone wins, really. Yep. Great. Well, thank you. It's fascinating to see the overview of a particular research project. We've got one more question in the audience. So,
I have one last question, and I guess I'm asking, hello. On behalf of our students at Alameda, many of our students are interested in the STEM career later on when they grow up. But as you know, it's quite competitive and it requires many years of study. Do you have any advice for our students who are about to commence their 11 and 12?
Yeah. What I would say is that, it's a really competitive process to get into vet school, but I don't think you need to put too much pressure on yourself. So there's a lot of people who come in via alternative pathways as well. So it's good to work hard in year 11 and 12 and do your best. And if you get in straight away, then that's great. But I think also thinking about the fact that getting some life experience and coming in via an alternative pathway is also an option. And so, not to think that this is the only way that you can get into veterinary science. We have a lot of people who do other things. They might work on a farm, they might do some vet nursing, and then come in and do veterinary science after that. And all of that experience, all that life experience, I think makes is really helpful as well. So it's not the be all and end all VCE.
Yeah, absolutely. Yeah. And so what subjects did you do in high school? And did you know from an early age that you wanted to be a vet?
So my dad was a vet.
Okay, yeah.
So I was very interested in veterinary science. I was always interested in science, actually. And I think that's how I ended up in a research career, is that I was always very interested in science and doing research. And but I did maths and chemistry and physics, the kind of straight sciences pathway, but it's not the only way. So,
absolutely, very good. That might help for some subject choices that are coming out for a lot of students. Okay, thank you for the question. That's great. Okay, so a couple more online. So Lalitha, will creating more vaccines be a way to overcome antibiotic resistance?
Yeah, definitely. That's a great question, and it's a really important thing. So one of the reasons that we don't use much antibiotics in our chooks in Australia is because we have amazing vaccines. And some of my colleagues at the University of Melbourne have been really involved in creating those vaccines for use in chooks in Australia. And so most of the chicken that you buy from the supermarket in Australia actually has never been treated with antibiotics. And that is 100% a result of amazing vaccines. And so creating new vaccines is a really great way of reducing reliance on antibiotics.
Yeah. Okay, great question then. And I guess following on from the vaccines, so how effective are the vaccines in reducing the need for antibiotics? I guess it's connected to what you just said.
Yeah, so most of our vaccines that we use in pets are really good. So we vaccinate for a few different diseases in dogs and cats, and they're pretty effective. Some of the vaccines for use in people, like if we think about the influenza vaccine, it's really good at protecting against the strains that you are vaccinated for. I was vaccinated for influenza this year, and I got the flu. And that's because the scientists who develop these vaccines are kind of guessing about what strains are going to circulate in Australia over the winter and when they're designing the vaccine. But if there happens to be another strain that comes in, clearly that happened to me, then I'm not protected against that vaccine. And so it's not perfect, but it's kind of the best we've got at the moment. So some vaccines really, really good. Others not 100%.
Yep. Fair enough. I hope you've recovered from your flu. That doesn't sound ideal. Another one from Holly, actually. Thank you for another one, Holly. Should we be giving our pets probiotics?
So probiotics is another really great question. So probiotics are healthy bacteria. So they're the good bacteria that live usually in your gastrointestinal tract, in your gut. And having a healthy population of bacteria in your gut's really important. We used to, not really think about the bacteria that were living in your gut, but now we know it's associated with your mood, it's associated with your immune system. It's really very amazing that how these bacteria interact with our bodies. But there's not a great body of evidence that looks at how effective probiotics are at returning to your population of bacteria. And there is a big variation in the quality of probiotics. So I'd say the jury's still out on that one.
Okay, there you go. More work to be done, as always. Very good. Okay, one from Grace now. Some people advise to finish the course of antibiotics even after you feel well. Others say doing this could produce antibiotic resistance. What are your expert thoughts on this?
Yes, this is something that we've thought a lot about, actually, because as you saw in the presentation, the longer you're exposed to antibiotics, the more likely you are to develop antimicrobial resistance. And so we really would like that if you are sick and you need an antibiotic, that you take it for the shortest amount of time possible. And so this mantra, everyone was always told to finish the course, but actually now we're thinking about how short a period can we treat people or animals with antibiotics for, and they would still get better. But what I would say is that your GP or your vet is the best person to make those decisions. And so you guys should take antibiotics for as long as you're told to take them. But your vet and your GP should be thinking about the shortest possible duration. And sometimes we know now in human medicine, that's one dose. One dose of antibiotics is enough. Or 3 days might be enough. Whereas in the past, we might have treated for a week or 2 weeks. So we're really cutting down the durations of antibiotic treatment.
Yeah, interesting. Gosh, we're just learning new things. I need to talk to my GP a little more then. Okay, we've got a couple of questions, but I know we've also have the experiment in the room that we want to go to as well. So maybe one more question, then we'll head to that. Okay.
Great.
So from Emily, can bacteria hold more than one resistance at a time?
Yeah, they sure can. And in fact, some of the when we talk about antibiotics sharing resistance, they do that using what's called a plasmid. So that is a bit of DNA that looks a bit like a circle that lives not in the genome of the bacteria, so not in its DNA, but kind of next to it. And those plasmids, those circles, are kind of really good at accumulating. They like to pick up new resistance genes. And so they might pick them up one after the other, and you end up with a little circular bit of DNA that is resistant to maybe 5 or 6 or 7 different antibiotics. And then if I pass that on to the bacteria that are around me, then we very quickly can end up with what we call pan-drug resistant infections, which is where the bacteria are resistant to every antibiotic.
Oh, okay.
Bad news.
Yeah, super antibiotic resistance. Gosh. Okay. Well, we might just pause there for the questions. So do you want to go over to what is happening in the room with the experiment and how can I help?
So we had our pets that we handed out at the start, and our pets were covered with bacteria that we can see with the blue light. So
should I collect a couple that we can test? Okay, maybe
if people want to have a look and see how much bacteria were transferred from their pet.
All right, so that's...
So I can already see some on Julia's hand, even though she just touched our pet here.
Oh, so I'm looking at the hands, not the pets. Oh, pardon. Right. I thought looking at the pet.
So,
okay.
And also, I bet if we have a look over here, we can also see it. Oh, look at this. So much bacteria on the bench.
Yeah.
From where people were touching the bench after they touched their pets. So
maybe over here?
Over here?
Yeah, yep. Who's been holding...
Who's been holding a pet? Do you want to have a look at your hands?
Oh, can I look at mine?
Yeah. Look at all that.
I haven't held one, but...
You haven't held one?
Hold your hands up so the camera can see it as well.
Oh, yep. A little bit on your thumb there, not too much though. Did you have pets inside or pets in the bed? Pets inside had less bacteria on them than pets in the bed. So, some there. Oh my goodness. So the other thing we can do as part of this experiment is you can go and wash your hands over there in the sink, and we can see how good you were at washing your hands. Oh, yep, you've got plenty.
So, Laura, I've been holding one for a minute.
Okay, let's see.
Let's see what happens. Now, I believe this was the one that sleeps on the bed.
Sleeps on the bed. Oh, my goodness.
Okay. So I definitely need to wash my hands. I think is what you're saying.
You definitely need to wash your hands. Great. Yeah. So you can see from this experiment that just really simple, behaviour like patting your pet can lead to accumulation of bacteria. But we not only get bacteria from our pets, we also give bacteria to our pets. And so that is a risk for our pets as well, particularly if their immune systems are a little bit compromised. So
amazing. Do you want to tell us a little bit more about what's sort of on our hands and how that all works with the UV light?
Yeah, so this is called Glow to Show powder. And it is glows in the UV light when you shine the UV light on it. So it's a really good way of, the other good experiment now is to wash your hands and then recheck, and that shows you how good you are at washing your hands as well.
So 20 seconds.
What's the
song you're supposed to sing? Happy birthday. Happy birthday.
Yeah.
Yeah, very good. Sing happy birthday to yourself every single day.
Yeah.
Okay, great. Well, we've got sort of 10 or so minutes, so a little bit more time for some questions. Anything else about that experiment or anything else, any advice you'd like to receive? So pop up your hand. We've got a few more questions online. Okay, so from Reva. What's your top advice for the public to help stop the spread of superbugs? I know you mentioned a few things at the end there, but maybe we can go over a couple of extra ones.
Yep. So I think the first one is to get vaccinated and vaccinate your pets so that we can reduce the amount of antibiotics that we use in the environment. And then also to really think about when you have a cold, whether you need antibiotics. Probably you just need to take it easy for a few days and do some TLC, and you don't need antibiotics. So thinking about that when you go to see your GP when you've just got a cold or a virus. And then the third thing is to wash your hands. So particularly if you've been touching animals that have been treated with antibiotics, if you've been out on a farm, for example, then it's just a really good practice just to wash your hands when you come inside, particularly before you eat. Yeah.
And sing happy birthday every single time.
Sing happy birthday, that's right. 20
Amazing.
seconds.
Yeah. Okay, great question. Okay, so another one from Beck. So do you think disinfectant surface sprays should be limited to use in clinics and hospitals rather than generally available? I guess that's trying to clean up too much.
I mean, bacteria are around everywhere. They're on every surface. And so using a antibacterial spray on the surface is really a very short-term thing. The bacteria are going to be back there really quickly. And we shouldn't be scared of bacteria. Bacteria are part of our world. You know, they're not inherently, the bacteria that are on our surfaces are not inherently dangerous, but particularly after doing things like handling raw meat, that sort of thing, then you definitely would want to, clean those surfaces really well using a detergent or something like that. Because those are the bacteria that can hurt us. The bacteria that are present right here on this surface, not going to be, very likely to be dangerous. So,
okay.
Yeah.
Very good. Okay, so one from Ellie now. We did touch on this in terms of going to see the vet. So what should I do if my pet is sick and the vet prescribes antibiotics? I assume it's listen to your vet, but what other advice would you have?
Definitely listen to your vet. They're the expert here.
Yeah.
If your pet needs antibiotics, you should definitely give them to them. We don't want our pets to be at risk. Because we, are scared of using antibiotics. If your vet thinks your pet needs antibiotics, then definitely give them. Same goes for when you go to the GP. If they say that you need antibiotics, we definitely want to take them. We don't want anyone getting super sick because they're scared of antibiotics.
No, absolutely. Very well said. Okay, one from Grace now. So does higher exposure to bacteria, e.g. kids living on farms, handling soil animals, produce lower rates of allergies? Interesting.
Yeah, interesting. Not my area of research, but yes, definitely the research would suggest that kids living on farms and kids that have pets at home do have lower levels of allergy. Yeah. So, super interesting. Yeah. I know there's a lot of people doing research on that, but not my area of research.
Right. Very good. Okay, no worries. Well, I guess coming back to your research and your job as a researcher, do you want to maybe talk a little bit about what just a general day or maybe a general week is? I know you spoke a little bit about the horse research, but no doubt that was over quite a long period of time. But general week, what would you be doing?
Yeah, so I do quite a lot of what we call, I call computer-based research. So I am really interested in how we can use data to change the way that prescribers, vets and doctors use antibiotics. And so I do a lot of research looking in big data sets, so looking at electronic medical records from animals, from 4 or 5 million records, which is all like on my computer and building models, and I really like that sort of stuff as well. And so I do a bit of that. I also have about 10 PhD students who I work with, so I do a lot of meetings with them to talk about what their research is like, and talking to my colleagues. We have quite a big research team. We have 30 people working in our group at the moment. So about the research they're doing, because we do research not only in Australia, but also in Southeast Asia and the Pacific as well.
Yep.
So, yeah.
Very interesting. We've
Yeah.
spoken to a lot of our presenters throughout the week about, all the collaborations they have across the world. So
Yeah.
are there particular areas that are doing similar research or that you work specifically with across the world?
So we do quite a bit of work in Southeast Asia and the Pacific about upskilling people to both look for antimicrobial resistant infections, but also to improve the way that prescribers use antibiotics in clinical situations. So
Yep.
we talk to veterinarians and to other prescribers about the common reasons why people use antibiotics when they're not needed, and think about ways that we can change people's behaviour, which
Yep.
is super interesting,
Yep.
very different to other areas of science, but
Yep.
really important as
Absolutely.
well. So-
And a lot of travel for you as well then, Southeast Asia?
Yeah, I don't do so much travel myself,
okay.
but our team does
Yep.
quite a lot of travel.
Yeah.
Yeah,
And a large team too. I was about to
Yes.
ask that, but a
Yeah.
team of 30.
Yeah.
It's,
Yeah,
it's a
it's a
lot
big
of
team.
collaboration.
Yep.
Amazing.
Yep.
Okay, so time's running out. So my final question, I guess, to you would be, there has been, a few questions of things that we still want to be finding out. But what is next for you? What's sort of on the horizons? What are you hoping to look at in the next couple of years?
Yeah, so what I would really like to do is to set up a antimicrobial use and resistance surveillance program is what I'm really interested in doing. And so in Australia, we have one for people, but we don't have one for animals. And it's a really great way of collecting data that to drive decision making.
Yeah.
So setting up surveillance is kind of the next horizon.
Amazing.
Yeah.
That sounds incredible and very much needed right now. Fantastic. Well, thank you so much for your presentation, for answering all the questions, for all your insights. It's been fascinating. Thank you, everyone, Alamanda College, for joining us today and taking part in our experiment. I know we all need to start washing our hands too from all that, the glow germ. And everyone, thank you so much for joining us as well. This has been our final masterclass, but thank you so much for joining us for today and possibly for the whole week. I hope you had a great science festival, and we'll see you again next year. All the best.